The next portion of Chapter 3 that we're going to go over is Concept 3.5, and that's proteins. Proteins include a diversity of structures resulting in a wide range of functions. So about 50% of the dry mass of most of our cells is protein. 50% or so of most bodies should be made up of proteins. Protein functions are more than just giving us movement as far as muscle goes and strength, but it also includes defense, storage, transport, cellular communication, movement, and structural support. I'm going to show you a whole bunch of videos that show you different ways that proteins work. In your textbook, you have a table that explains each of these proteins also, but I think this is kind of a good way to show you what each of these proteins do.
Contractile proteins, like the motor protein shown here, can move parts of a cell. Other contractile proteins, working together in muscle cells, can move a whole animal. The immune system makes defensive proteins called antibodies that bind to invaders, such as the virus shown here, and mark the foreign objects for destruction. An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
So one thing to know about enzymes act as catalysts, which means that they speed up chemical reactions. They're never consumed by the reaction though. As soon as what binds to it gets spit out, that enzyme is still there to make a reaction happen later on. And basically, if we didn't have this super duper important protein, we would not exist in life.
Gene regulatory proteins bind to DNA in particular locations and control whether or not certain genes will be read. This allows cells to become specialized for different functions and respond to changes in their surroundings. Signal proteins include hormonal proteins that help coordinate an organism's activities by acting as signals between cells. For example, insulin, a hormonal protein secreted by the pancreas, signals in animal cells to take in and use sugar. The hormone receptor is also a protein. Receptor molecules bind to signal molecules and can then emit second messengers which trigger changes inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules, such as hormones, are also proteins. Sensory proteins detect environmental changes such as light and respond by emitting or producing signals that call for a response. Storage proteins stockpile building components that cells can use to make other proteins. Storage proteins in seeds provide raw materials used by the developing plant, unless an animal eats them first. Ovalbumin, the main substance in egg white, serves as a storage protein for developing chick embryos. Storage proteins stockpile. Structural proteins have many functions. Like tent poles and ropes, they shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins. Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell.
Okay, so when we were talking about carbohydrates, we said that the monomer of a carbohydrate is a monosaccharide, right? Build a bunch of those together and you have a polysaccharide and you have a polymer. Well, the building blocks of proteins are things called amino acids. Amino acids are organic molecules that have a carboxyl and an amino group, and then they also have these side chains that have very different R groups that they can have, and that's what makes each amino acid different. Polypeptides are unbranched polymers built from the same set of 20 amino acids. And a protein is a biologically functional molecule that consists of one or more polypeptides. So a polypeptide is a polymer, but a protein is also a polymer too, and a protein is larger than a peptide. So amino acids are the monomers or the building blocks. They make up a polypeptide, and then a bunch of polypeptides will make up a protein. You do not need to know the R groups for amino acids. Just know that there's a number of different R groups that they have, and that's what makes amino acid different. It depends on which little side chain or R group. Hydrogen here makes a simple one, CH3, these are a little bit more complex. So there's different side groups, and I think there's 20 or so of these, I believe. What I would know is that they are charged differently. So there are some side chains that make the amino acid negatively charged, and some that make them positively charged. And so basically, the reason for knowing this is that the interactions between those two make proteins have these really complex shapes that will make more sense when I show you some videos later on. So amino acids are linked together by peptide bonds. Just like with carbohydrates, they were glycosidic bonds, and with lipids, it was triglyceride linkage. Well, in this case, it's a peptide bond that links amino acids. Polypeptides range in length from a few to more than thousands of monomers, and each polypeptide has a unique linear sequence of amino acids, and that sequence gives it these really unique shapes that make proteins look like how they do. So once again, you have a peptide bond that binds two amino acids together. In order to make that peptide bond happen, we have to have a dehydration reaction where the water is released, taken out, so that we have room to make that bond. Say that we want to split this up again, you add water again, and then it will split up. And that would be a hydrolysis reaction to break up polypeptides into just amino acids. So a functional protein consists of one or more polypeptides that are twisted and folded and coiled into a very unique shape. And here's just kind of an example. You'll see the ribbon model a whole lot, and then there's that space filling model, and every protein looks a little bit different. That's why blood cells have the specific shape that they do have, where it's almost like an inner tube, has like a donut kind of, but without the actual hole in the middle. So the amino acid sequence of each polypeptide leads to a protein's three-dimensional structure, and their structure leads to its function.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
Okay, so this end of this image right here, all these little circles, these purple circles are amino acids. They're connected together by polypeptide bonds. How did that polypeptide bond form? Through a hydration reaction, right? Where you added, a dehydration reaction where we added water to allow that peptide bond to form. The way all of these little R groups that come off of each amino acid lead to a certain shape because they repel each other or they're attracted to one another. There were some that had a negative and a positive charge, right? All proteins are very diverse. There's lots of different types of proteins within the body and within the cell. You learned about transportation proteins and structural proteins and enzymes, etc. But all proteins share 3 levels of structure: primary, secondary, and tertiary. And then the 4th level is the quaternary structure. So primary was the image of all the amino acids. Secondary, well, let's move on and look at it. You'll see some good pictures of it. So primary structure is unique to the sequence of amino acids. It's like the necklace. Secondary structure is found in most proteins. It will, depending on how those amino acids interact, the R groups interact with one another, they either coil or they make like a kinky fold. like like the 80s hair, whatever that was called. And then tertiary structure is determined by the interactions among various side chains. Quaternary structure results in interactions between multiple polypeptide chains. So here we go. Primary structure is just all the amino acids linked together through peptide bonds. Here we go.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
The secondary structure will make either a beta pleated sheet, the little kinky sheet, or an alpha helix, a coil.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
And then the tertiary structure is how all those secondary structures kind of respond to one another.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
And then quaternary structure are all of those tertiary structures binding and being attracted to one another to make a type of protein that we see.
Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
So he says quaternary structure. I have always said it quaternary, but you can say it however you want, just as long as you know what it is. So to kind of further talk about the red blood cell and the importance of the structure of a protein and how it makes things happen, that hemoglobin molecule, you saw those 3 those 4 tertiary structures together kind of lead to a little bubble or a little divot inside of a red blood cell. Well, some people have this inherited blood disorder where there's one amino acid that changed, that has been substituted from a regular hemoglobin. And as a result, your red blood cells don't look quite correct. So this is what a red blood cell should look like. You have all of these tertiary structures binding together that make this little, hole, little seat inside of a red blood cell. One little teeny tiny change, just one amino acid is changed in the beta units, I guess, and your red blood cell takes on this kind of half moon type shape, a sickle shape. And as a result, if you have sickle cell anemia, you are unable to bind oxygen very well. Exhaustion is very, very common. It's very painful to have sickle cell anemia. It's a pretty horrible, terrible disease to have it. And it's all an inherited trait that results in just one amino acid that has altered the entire shape of every single red blood cell in your body. So what determines protein structure? In addition to the amino acid sequence, there's also physical and chemical conditions that can affect protein structure. You know this when you straighten your hair or when you curl your hair. Heat changes the structure of the protein of your hair. So you can change the pH, change the salt concentration, change the temperature, or there's other environmental factors that will cause a protein to unravel. The loss of a protein's native structure is called denaturation. It is not usually permanent. A denatured protein is inactive, but usually it can become renatured. You have really curly hair, you straighten your hair, you've denatured it. You wash it again, the heat is gone, and now it's renatured back to its original state. So a normal protein can become denatured, but it can also be renatured. The last carbon-based compound that we are going to talk about in macromolecule and that you need to know and that exists are nucleic acids. Nucleic acids are really important molecules. They store, transmit, and express hereditary information for us. Without nucleic acids, we wouldn't have DNA. So the amino acid sequence of a polypeptide is programmed by a unit of inheritance called a gene. So the amino acid sequence that makes our cells the way that they should be are made that way because a gene tells it to do that. When you have sickle cell anemia, you have a gene that says replace that one amino acid. Genes are made up of DNA, a nucleic acid, which is a nucleic acid made up of monomers called nucleotides. So nucleotides are the monomers. DNA is something called a nucleic acid, diribose nucleic acid. And the monomers that make up this macromolecule called nucleic acid is called a nucleotide. There's 2 types of nucleic acids. There's DNA, deoxyribonucleic acid, and then RNA, ribonucleic acid. So DNA provides directions for its own replication, and then DNA also directs the synthesis of RNA, messenger RNA, and messenger RNA controls protein synthesis. So DNA gives the information to the messenger RNA. Messenger RNA leaves the nucleus and it goes and it makes a protein, tells the protein what to be made. So we have DNA, our strand of DNA makes a messenger RNA. The messenger RNA leaves our nucleus and gets into the cytoplasm of the cell, the guts of the cell, and it comes over to a ribosome and it tells that ribosome how to make amino acids. And those amino acids, as we know, become a polypeptide, which becomes a protein. Nucleic acids are polymers called polynucleotides. So a nucleic acid is a polymer, yes. Another name for a nucleic acid is a polynucleotide. What is the monomer of a nucleic acid? Amino acids, right? Each nucleotide consists of a nitrogenous base, a pentose sugar, and 1 or more phosphate groups. Portion of the nucleotide without the phosphate group is called a nucleoside. So if you, well, let's see. Here, I want to show you. This is a nucleotide right here. So you have a phosphate group, a pentose sugar, and a nitrogenous base.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide.
Okay, I'm going to stop that video there. So that was a lot of information that we haven't really covered yet, right? But we did say that each nucleotide consists of a nitrogenous base, a pentose sugar, and 1 or more phosphate groups. Each nitrogenous base has 1 or 2 rings that include nitrogen atoms. And so it's either going to be a pyrimidine or a purine. Pyrimidines are cytosine, thymine, and uracil, and purines are adenine and guanine. So I don't really have a great way to remember it, except that pyrimidines, even though it's a long word, pyrimidines, even though the word is longer, is only made up of 1 circle. Purines is made up of 2 carbon circles, even though it's a shorter word. Thymine is only found in DNA, and uracil is only found in RNA, and the rest are both found in both DNA and RNA. So here's our strand of DNA. Another thing that the video, I stopped it before it really got too much into it, is that we have a 5' end and a 3' end. So you can't just flip a DNA strand over. They have to be going the correct direction to link. So there's actually, on a DNA strand, there's 2 of those. There's 5' end and then the 3' end, and the other side will be opposite, and they will link. And they'll end up twisting and making the double helix that you know is DNA. If you remember from Jurassic Park, the original Jurassic Park especially, they had that little, I think he was like a DNA cartoon character, and he was that little double helix. So we have our strand, we have our sugar phosphate backbone. So this part right here is twisted, and it's now the sugar phosphate backbone, 5' to 3'. And then sticking out on the outside are these nitrogenous bases. The nitrogenous bases are either a double carbon circle or a single. Now, what's interesting is that they will link with one another. A purine can only link with a pyrimidine. A pyrimidine can only link with a purine. They cannot link pyrimidine to pyrimidine and purine to purine. And the way that they go is adenine has to link with thymine and cytosine has to link with guanine. The 2 curved ones have to link together. The A and T have to link together. When that DNA molecule makes an RNA molecule, the thymine is replaced by a uracil. Uracil is only found in RNA. The sugar in DNA is deoxyribose and in RNA is ribose. The prime is used to identify the carbon atoms in the ribose, such as the 2' carbon or the 5' carbon. A nucleoside with at least one phosphate is attached. So nucleotide, we already kind of talked about that. So you have your backbone, sugar phosphate backbone. Sticking out are your nucleic acids, and there will be another strand that will go 3' at the top and 5' at the bottom, and it will link these nucleic acids. These nitrogenous bases will link with one another, making it twist and do that double helix. Okay, so adjacent nucleotides are joined by covalent bonds on the 3' carbon of one nucleotide and the phosphate of the next. So the phosphate of one and then the 3' carbon of this one. So the phosphate and the carbon hold hands. And those links together will form the backbone of that sugar phosphate unit with those nitrogenous bases sticking out as little hands. And then the sequence along the DNA or messenger RNA is unique for each individual gene. RNA molecules usually exist as single polypeptide chains, so that's a single chain, and DNA molecules have 2 chains together, and they end up spiraling and forming the double helix. In the DNA double helix, the 2 backbones run in opposite directions. I mentioned this before, right? So it's 5' to 3. On the other side, it's 3 to 5. And that is called being anti-parallel. So I would know what a double helix is. I would also know why DNA is considered anti-parallel. One DNA molecule includes many, many genes. And so here you can see that double helix forming. We have the 5' snaking down on this side, and then the 3'. So it's 5 to 3, and then 3 to 5 at the bottom, right? These the hydrogen bond, these base pairs, nitrogenous bases, will hydrogen bond to one another. AT always, CG always. And the way that they hydrogen bond with one another forms the curving that makes the DNA have this twisting pattern. When DNA turns into RNA, it will unzip itself and form a new strand in the middle, and then it will zip back together again.
DNA consists of 2 complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the 2 sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine.
Nitrogenous bases in DNA pair up, form that hydrogen bond. Adenine is always with thymine, guanine is always with cytosine. And that's called complementary base pairing. So when you're asked, what does that complementary base pairing mean? It's where the DNA, the nitrogenous bases on the DNA are always going to pair up with one another, always AT and GC. And then in RNA, thymine is gone and it's replaced by uracil. So it's AU